This is Yukio Nagasaki from University of Tsukuba. I'm happy to present here. Let us start my presentation. What I'd like to talk about today is how our nanoparticle-based antioxidant work for radiation therapy, which has done these peoples. We have no COI to open. Let me show you oxidative stress and antioxidant. However, due to time, I put the explanation in YouTube. If you have a time, please visit here and here. But briefly, conventional antioxidant dysfunction of intracellular redox reaction. This is a problem. Nanoparticle antioxidant prevent the dysfunction of intracellular redox homeostasis. This means, less toxic effect of nanoparticle-based antioxidant. Capture this and see video on web. So this is the explanation of enhancement, permeation, and retention effect. Due to the leaky blood vessel in tumor site, the large molecular weight compound, including nanoparticle, permeate very much easily. However, the immature lymphatic system, these kind of high molecular compound nanoparticle retain here to improve the concentration of nanoparticle itself in tumor site, which is known enhancement, permeation, and retention effect, which was proposed by Professor Hiroshi Maeda. Unfortunately, he passed away last week. So today we are using nanoparticle antioxidant for radiation therapy. We inject the antioxidant nanoparticle prior to irradiation. As I mentioned, from the EPR effect, injected nanoparticle tend to accumulate in tumor site and scavenge reactive oxygen species there. So what happened? Question is, does RNP, our nanoparticle, accumulate in tumor site and affect the radiation therapeutic effect? As you know, radiation therapeutic effect, let me show you briefly. There are two different effects, indirect effect and direct effect. Direct effect causes the cleavage of DNA by direct irradiation. Which is around 20 to 30% causes apoptosis. Indirect effect generate reactive oxygen species and loss, damage or cleave the DNA, which causes necrosis. Actually, 70 to 80% of the radiation is caused by indirect effect. At the same time, generated loss spread to whole body and causes adverse effect like this. So we wanted to eliminate this kind of loss in the whole body. However, at the same time, we may decrease loss in tumor site. So the question, RMP is anticipated to suppress the adverse effect like this. But how does RMP work against therapeutic effect here? So initially we thought suppression of necrosis and selective apoptosis occur, which is much help for increase the QOL of patient, although the efficiency might decrease. So this is why we started this project. So first, we show you effect of self-assembling antioxidant nanoparticle RNP against the whole body radiation. The, uh, the radiation protector have many applications like this. We have several, radio protector. One of them is amifostin here. However, this after administration, phosphoric acid cleaved by alkaline phosphatase and become antioxidant. So this means low molecular antioxidant causes the side effect. Another drug is peripherin, human keratinocyte growth factor. This is again very strong adverse effect like this. So protective agent must not give side effect because we need to inject them to healthy people. So then we wanted to check our nanoparticle antioxidant to healthy mice 24 hours prior to irradiation. So this is data of 7.5 gray, which is equal to 7.5 sievert by gamma rays. And it is more than lateral dose of human. As compared to control, 7.5 gray irradiation shows, the bleeding of all organ like this, which means generated loss circulate in bloodstream and attack the organ like this and start bleeding. In case of low molecular antioxidant did not decrease the bleeding like this, but in our nanoparticle-based antioxidant did not show any bleeding like this. This is data of 6-gray exposure without antioxidant, only irradiation. After 1.5 years, all of mice died like this. In case of low molecular antioxidant did not cause the significant effect. However, in our nanoparticle-based antioxidant, although 20% die, but still 80% survive. So this means less toxic of antioxidant and also long-term circulation because we irradiated the mouse after 24 hours after injection. So we confirmed RNP was show radioprotection effect. Then we move to therapeutic effect. How does RNP work for radiation therapy? In this case, we irradiated the tumor-bearing mice. So after injection of RNP, so we used the RNP right here and amifostine as a control, commercially available radioprotection agent. We used twice dose compared to our nanoparticle and then show the efficiency. This is tumor growth profile as compared to control without irradiation. Irradiated the tumor, suppressed tumor growth. This is saline control, amifostine and RNP. We are amazed to see, even though we administer RNP, which accumulate in tumor scavenge reactive oxygen species, but tumor growth suppressed significantly, rather higher efficiency as compared to saline. So this is the adverse effect. You can see here the body weight increase in the case of healthy and tumor-bearing mice without irradiation. With irradiation, although their body weight decreased for several days due to the stress of radiation, but the saline control profile like this, amifostine and RNP, the body weight of RNP-treated group recover like this. This means less toxic, toxicity decreased. Finally, this is survival. Without radiation, the profile like this. With radiation, saline, amifostine, and our RNP. RNP administration prior to the X-ray radiation extend the life more than half. So we clearly show the very high effect for radiation therapy. And next, we confirm the adverse effect. Blood cell, although red blood cell and white blood cell did not show any difference, but the platelet, by radiation, the number of platelets decrease, but the RNP recover like this. Kidney function, the creatinine and BUN level increased by radiation, but the RNP suppressed like this. Liver function, LT, ST, TBIL, increased by the irradiation like this, but the RNP again suppressed very much. The RNP treated group effectively protected damage to blood cell and organs. This is histology surrounding a tumor due to time. Just I mentioned, the RNP treatment group similar to saline group. The irradiation without antioxidant amifostin causes strong damage like this. This is intestine. By irradiation and amifostin shows very strong damage to intestine, but RNP suppress damage. Quantitative data shown in like this. So finally, we are thinking the mechanism. The radiation causes loss and which suppress, which attack to DNA. As I mentioned, this is indirect effect. But at the same time, loss is influenced very much around, the tumor microenvironment. Activate NF-kappa, B, TNF, alpha tissue factors, which prevent the therapeutic effect. But our nanoparticle antioxidant treatment confirmed to suppress the activation of these factors. This means, the resistance of tumor is decreased by our RNP treatment. Then we attack, we irradiate by X-ray. Then the therapeutic efficiency did not decrease. Finally, let me show you in vitro experiment about apoptosis. When we irradiated the cell in the presence of RNP, the green is apoptosis level. This is clearly shown as compared to other. The ratio of apoptosis increases by treatment with RNP, which is, the patient-friendly treatment. In conclusion, self-assembling antioxidant did not decrease therapeutic efficiency even by the pre-administration. The RNP pre-administration suppressed damage of normal tissue and organ caused by the radiation treatment. Therefore, the lifespan is significantly extended by the RNP treatment pre-administration. The RNP pre-treatment for cancer radiation therapy improved the quality of life for patients. Thank you very much for your kind attention.